Okay, well, welcome. I'm talking today with Dr. Megan Falsetta. Dr. Falsetta is a professor at the University of Rochester Medical Center, where her research focuses on causes of pain in patients causes of gynecologic pain, including vulvodynia and lichen sclerosus, which is a skin condition that affects the vulva. She also looks at the way that bacteria and yeast interplay with these conditions to either start them or worsen them or can be addressed to treat them. So I'm very excited to talk with Dr. Falsetta. Dr. Falsetta, welcome.
Thank you.
So your lab has so many projects going on, and I am fascinated by all of them. So how did you get into this particular space of research?
So I was going to be a microbiologist and I was going to study lung biofilms, but the universe had something else in plan for me. So I came to the University of Rochester originally on a postdoc to study biofilms in the oral cavity and how collaboration between yeast and bacteria accelerated cavities in young children. And then when that was up, I found another postdoc position working on a disease I'd never heard of, vulvodynia. And immediately I was struck by the fact that, first of all, this was nowhere in my user's manual. Why had not anyone told me that this could happen to me or what I would do if it happened? And then that kind of opened my eyes as I got more involved with our gyn colleagues about the wide variety of diseases that can affect the gynecology in the gynecology world that can affect pretty much any reproductive organ that women or people with these anatomical features know nothing about. And so really that kind of motivated me to start to understand better the mechanisms of disease because we really don't understand them very well at all. And so it's just kind of expanded and someone will pitch an idea my way, or I, fortuitously will collaborate with someone where we can get waste tissues or something that gives us an opportunity to study something new. And I'm usually ready to give it a try. So that's how we've kind of come to doing so many different things. We work a little bit with endometriosis, vulvar lichen sclerosus, as well as our, bread and butter and longest standing research has been in vulvodynia. But hopefully we'll be able to branch out with, if the NIH is able to fund the research, then we'll be able to do a lot more things.
It's very exciting. So the thing that caught my eye was something from the National Vulvodynia Association about a medication for vulvodynia. And the medication is called, I think it's called Miricin 1. Is that right? Am I saying it correctly? I don't know if there's yeah.
Yes. Yeah. Some people say Miricin, some people say Miricin, but it is a specialized pro-resolving mediator. So your body naturally takes dietary fatty acids, particularly the reason why you hear so many good things about how you should consume fish oil, because it's rich in things like DHA, and this helps to make these mediators that aren't like steroids or like Tylenol, ibuprofen, your nonsteroidal anti-inflammatory or NSAIDs. They help to resolve inflammation without completely blocking it. So they allow the process to happen, but they get you back to that just right, happy middle faster and more efficiently. And so we have reason to believe in vulvodynia based on a lot of data we've collected that there is a dysfunction in this lipid profile in the vestibule, in that painful area in patients with disease, and that supplementing back topically, at least in a mouse model, in an in vitro model with cells, seems to be a very promising pathway forward. So the goal would be to develop this Miricin into a topical application, and we are currently working with a drug company that is interested in doing just that. And we have a use patent, so we have a specific patent saying this was a unique idea to use this particular drug for vulvodynia. So it's a naturally produced thing. Your body naturally makes it. It's safe. It's effective at very, very low doses. But of course, once we make it in a lab, it now becomes a drug. So it has to go through rigorous safety testing. And so that's where we're embarking, hopefully in the near future, to test and make sure that it is in fact going to be safe. And then if it passes the first hurdle, then we start to work towards seeing if it actually works for the disease.
Fantastic.
So it's a long road to go, but I think there's a lot of promise. And, we've seen other labs work with the compound and get similar results, that it has the ability to relieve pain and is very promising in the arena. So I'm optimistic, cautiously optimistic.
And so I'm curious about what your thinking is for the application, because obviously we see people with general vulvar pain, but mainly we see people with vestibulodynia, pain in the vestibule specifically. And so do you think it could be used for broad applications for, because obviously we think that there are people who have, neuroproliferative causes of their pain in the vestibule, too many nerve endings related to things like mast cells and underlying disorders or provocation from things like yeast versus, like hormonally mediated vestibulodynia, which we tend to treat with hormones. And probably I would think that this would be more for the former, not the latter.
Yeah, I think it's hard to say because we really don't know if those are just clinical groupings, whether there's real mechanistic research behind the different subtypes of vulvodynia. And some agree that there's a neuroproliferative version. Some are less invested in that hypothesis. What I can tell you is that the Muricin does more than limit inflammation. It also directly acts on pain channels, specifically the TRP channels involved in pain. And we've implicated in at least provoked vestibulodynia that those TRP channels are involved. And so it doesn't just work to bring down inflammation, but on the order of seconds will turn off that pain signaling in our in vitro models. So it's likely to have analgesic properties for any of these subtypes in which you have pain signaling happening. So we're looking at fibroblasts. They're not the nerve cells. They're not the ones necessarily conducting the pain, but we would believe that it would probably have application on the nerves in the tissue and help to slow down that pain signaling that's being conducted through the nerves. And others have seen this. It's not just our work. They've seen in other disease indications with similar compounds, not necessarily Muricin in some extents, but resolvins as well, which is like close cousin of Muricin. And they've shown the same sort of thing where they can very acutely stop pain signaling. So in the order of seconds, turn off that pain signal potentially.
Because
So I think it's going to broader application.
yeah, that's what I
and
was,
hopefully for generalized as well. I mean, I can't say for certain we don't have
sure.
samples from generalized patients. I can't tell you with good confidence that that's going to work, but I don't think it could do harm. So certainly, the initial indication that it might get approved for is localized vulvodynia or provoked vestibulodynia, whatever term it is that you prefer. So it's probably
Sure.
going to get approved there first,
Yeah.
but that's not to say that someone might not use it off label.
Yeah, because
And
when you're
similar compounds have been used for dermatitis as well. So
yeah.
there's a lot of application for these things.
Yeah, it seems like those pain signals or that pain signaling pathway is used in so many things. We were talking to the NIH in our group about a compound that can affect those channels, and they've been using it in applications like, arthritis. So it seems like there could be, many applications like injected intra-articularly, anything that would affect those particular pathways. It seems could be very broad.
And topical capsaicin has shown, repeat application of topical
Exactly.
capsaicin,
Yeah.
despite how unpleasant it is, can be used to treat provoked vestibulodynia.
Mhm.
And it's in part because what it is doing is affecting those channels. So it's overstimulating those channels to the point where the cells that make those channels are essentially killed off. So you get rid of the cells that are problematic, and it'll last for a while, maybe not indefinitely, but so this would be a non-painful, non-irritating
Right.
way to do the same sort of,
Which
hopefully
is
shut those
advantageous.
channels down.
Because I know that there have been studies with resinoferritoxin, which is, a capsaicin analog that's much more potent, and I'm sure would be quite painful. And so it's nice to think of something that would be, have that beneficial effect without the associated pain.
Yeah, it could potentially, I mean, it's short-lived. It's not going to be long-term sensitivity. So it could be applied topically, probably under, some form of local anesthetic. So, um, so it would depend. Obviously, you're going to have to choose your anesthetic carefully. Probably something injected is going to be better so that you don't put something that's coating the tissue to prevent the penetration of your resiniferatoxin.
Right. So for Muricin, I don't know why I'm having trouble with that. How long do you think that what's the half-life of the drug? Do we think? Like, how long will it last?
So Yeah, so that, I mean, that's been the issue, is that these things are turned over pretty quickly in the body, and
Mhm.
they are sensitive to oxidation, oxygen, but manufacturing under the right circumstances. So, I can't get into any, drug secrets,
Sure, sure.
but
We're not stealing
you
anything
know,
here.
basically, there's yeah, I mean, it's not even mine. It belongs, the drug company we work with, but basically slightly chemically altering these, which again makes them drugs, but altering them to make them more stable so they will last longer and not be as sensitive to these external pressures. So obviously the goal for anything would be to have at least a shelf life of maybe a year, something you can
Mhm.
keep in a drawer in your
Sure.
bathroom.
Yeah.
So that's the hope. That's the hope. So
Yeah.
things like, as drug development is ongoing, is there's a lot of stability testing that's being done to make sure that this will actually be something that will, it won't have a tube of it that'll last a week, that may, or have to have it compounded weekly for you. That would be very cumbersome and not effective. So, so there's some challenges there that have to be overcome, but they've definitely got a lot of answers to those problems already.
Sure. Yeah, because I think about our patients who use gabapentin, they have to apply something up to 3 times a day to get relief. And for capsaicin, it's at least daily. And so I'm also thinking not only about like shelf life stability, but like how often are people going to apply it? Because I know for people who have to apply something more than 1 time a day, it can be really challenging to adhere. And often I'll see people in the office who, they were getting benefit. Maybe they got 60% better, 70% better, but it just was exhausting. I take one medication and it's really, I have no side effects from it. And I do well when I take it and I don't do well when I don't take it. And it's still really hard for me to adhere to it, just a once daily medicine. So I know that it's hard for people. And so I'm wondering how long you think it'll last in terms of its effect.
Effect? Well, that's a good question. We really don't know yet. I can tell you from our mouse models is that we usually would treat them once a day,
Mhm.
Monday, Tuesday, 7 days a week. And they usually show improvement where their thresholds recover, where they're not showing as many signs of pain. They don't seem to be as sensitive to touch within as little as a week.
Okay.
And then that can be maintained for months afterwards, but I can't say with certainty that the model that we use fully recapitulates what we see in humans.
Sure.
Mice are not humans; they never will
No.
be. No matter how well we humanize them or what we do to them, they're never going to be human.
Yeah.
So it's not a perfect model, but when you put together multiple models, you can kind of, as I tease my students, there's not one model to rule them all. So you put the different models together, you get a more complete picture. So
Yeah.
I think that the way I suspect it would work, it would be very similar to topical steroid application for lichen sclerosus patients, where probably in the beginning, you may need to apply it more frequently, every day, twice a day, 3 times a day, whatever it is based on your personal needs, and that over time that the need to dose would taper off, where you might only need to administer once a week or every few weeks or when you have a flare up.
Mhm.
So that's the hope.
Mhm.
But I think that there is a genetic um, mechanism that underlies the disease. There's data to support that, and hopefully some of our future research, if we're funded, will look at the genetic underpinnings of disease. But
Mhm.
I think that there are some genetic mutations that occur that make the lipid processing um, dysregulated, and that there is no real permanent fix then for that. So you may need continual supplementation, but the amount that you need and how frequent may reduce over time, and it may just be periodic usage. That's what I'm hoping for. But of course, with the drug tests that will happen, there'll be a lot of different scenarios that will be tested to understand what the optimal dosing will be.
Yeah, it'd be so
So,
interesting to see what the genetics are, because it makes sense to me that if
and
you
it makes sense.
that there's a specific test you can do to tell if someone's predisposed to this, this is a medication they can have on hand. Because we see also a lot of the downstream effects from people who've had the pain and then they get pelvic floor muscle dysfunction. They may even get secondary pudendal neuralgia. And there are all these cascades of things that happen from the initial insult. And so being able to know that you're at risk and treat it quickly and not have some of those downstream effects would be amazing.
Yes. And so that is another one of our big goals. So the big problem with most vulvar and gynecologic disease is that we have no simple tests, no great tests. For lichen, it requires a biopsy, but you
Yeah.
have to have visibly affected tissue when the disease, from what we've gathered so far, probably starts years before you see a change.
Mhm.
Endometriosis, still the gold standard is a laparoscopic surgery for diagnosis to visualize lesions. And for vulvodynia, it's really expert opinion. So I think the stats that I've heard from our friends at Tight Lipped are very depressing, but that the average vulvodynia patient probably sees over a dozen providers and may spend up to 7 years or more before they get a diagnosis. And that often early on, one of the first things that they get told is just relax, maybe go home and drink a glass of wine and it'll all be okay. And that is definitely the worst advice you could give someone in that situation. It's only going to make it a lot worse.
Yeah.
To then
Yeah, we actually have a
also
paper
generate
that
the
is
alcohol.
we have a paper that's coming out soon talking about the rates among our patients of getting asked those questions and how distressing they are. And it's incredibly distressing for them to hear that. It's like my Katie Dumas, who's a fellow with Sarah Signa, graduating and becoming an attending at GW at the Saga Center, who's going to do this kind of work, was talking about how we never would tell someone who has back pain to have a glass of wine and, go lift something heavy. But for people with sexual pain, for some reason, we think it's appropriate, and it's not. It's traumatizing to the patients.
Yeah.
Yeah.
Yes, well, and it comes down to, Tight Lips' mission is that it really has to change at the provider education level, that some of this information needs to be required in medical school or other medical training programs at the, beginning level, as well as in residencies and fellowships. Like, this information needs to be available to everyone. So
Absolutely.
if providers are aware, if patients are aware, we will have less of that misdiagnosing or lack of a diagnosis and being given some really incredibly bad advice.
Yeah. Yeah, that's so true. Well, I'm curious about your mouse model and vulvodynia. And I'm curious, do you feel like it is, you feel like it's, and I guess this is because in my mind, I have such specific connotations for the terms, vestibulodynia and then like generalized vulvodynia. But can that mouse model be so specific? I'm just thinking about the anatomy of the mouth and how small these areas are.
Yeah. So for one, the vulva of a mouse is hair-bearing skin. It's not the same type of skin that you
Okay.
see in a human. It's
Yeah.
mucocutaneous.
Yeah.
So there are some limitations. There's not, a clearly delineated vestibule.
Right. Like the endodermal
so the
tissue.
anatomy is definitely
Yeah.
right.
Yeah.
Yes, and they can't tell us about the quality of pain. So we can watch things. So we're looking to improve our model by getting more behavioral measures in there. So, mice, when they're in pain, just like humans, show a range of behaviors like withdrawing from social situations or not doing the same activities they normally would do, or, showing stress behaviors. And so we can quantify those. So this is one of my graduate students' projects. She's very interested in understanding the kind of full spectrum of behaviors to see if this begins to mimic what we see in humans or not. So, mice are mice, and there's only so much we can do with a mouse. But I think as far as being another arm for doing testing, preclinical testing, so that we know, is there any hope that this might work in a human? I think it's a good pass through there. But I actually tend to have more faith in measures I get from human cells. So a lot of our work relies on primary human cells, cells that we take right from the patient. They're consented for the study. They know what they're getting into. We take biopsies when they're undergoing surgery, and we use those biopsies to grow human cells. And even most recently, trying to isolate the mast cells from that tissue to see what is special about those mast cells, and as well as potential epithelial cells and other cells. So really trying to come up with, and we're starting to dip our toe in the water with microfluidic systems, this idea of organ or tissue on a chip, with some collaborators we have in dermatology and engineering. So, I think we can do even more with the human tissue and the human modeling. And I think but it's still, in pain, it's very hard because there is no way to measure pain in something that doesn't experience pain, like an individual cell. But with our model, we have established that these inflammatory mediators, prostaglandin E2 and interleukin 6, can serve as surrogate measures of pain. So they're kind of biochemical measures that can be extrapolated back to patient pain thresholds. So we have that at least. So when you put all these pieces together and you get more pieces of evidence from each model system you look at that support that, I feel more confident that it's worthwhile to move into human trials. But the only way to really know if it's going to work is to try it in humans. But of course, it's expensive, it's time consuming, and people that enroll in those trials are taking a risk.
Yeah.
And so we want to make sure that we protect those individuals and that we don't waste time and expose people to, unneeded unknowns. So I think it's just one more model to pass it through. So once I get, positive from 3 or 4 different models and ways we've tested it, then I feel more confident. And with this particular application, I don't think there's any real risk for harm other than it might not work. We might waste someone's time when they could be getting another intervention.
Right.
So that would be my major concern. But the sad fact is that no matter what animal model you use, about 11% of those positive animal trials result in an actual drug that comes to market in humans. And there's many reasons why drugs fail. And one is mice aren't humans.
Yeah. So when you say, I heard you say sort of, the chip model sort of, so I'm guessing that's multiple kinds of cells so that they can interact together on a chip. Is that different than an organoid, or are they similar? Because I've heard talk of organoids
It's
like,
a little bit different because yeah, so it's a little bit different in the regard that usually there's biosensors employed. So you can have
okay.
membranes where you can have cells on one side that can talk to cells on the other side because it's a porous membrane. You can sometimes watch cells migrate from one side to the other. But the big thing that we're interested in is that this particular biosensor that they're working on allows continuous monitoring of interleukin-6, that surrogate measure of pain, where we can in
Right.
real time on a second by second basis know how much our cells are producing in response to different stimuli. And so that's where we'll start. And then, we can build up and get more complicated, start simple, one cell type, one
Sure.
measure,
Yeah.
and then build it up to being able to measure multiple with multiple cells. But
Okay.
it's an interesting thing that we're just starting to dip our toes in the water. It's a collaboration with another graduate student in
Yeah.
engineering who was looking for a great source of cells. And it's like, well, I've got all kinds of cells. So we're just talking about how we need to buy a new freezer because we're filling up our current freezer. So that's exciting that we have this huge library of strains, and we're collecting strains from lichen patients, from endometriosis patients, from patients undergoing gender affirming hysterectomies. So we have some
Yeah.
reference tissue or undergoing hysterectomy for other indications. We've got tissue from our vulvodynia patients that's consented, and that study is ongoing, as well as we've been getting tissue from scar tissue from colleagues in plastic surgery. They remove a lot of keloids in hypertrophic scars to see if that scarring process looks similar to the quote unquote scarring process that we see in lichen sclerosus.
Yeah.
So yeah, so we're that's basically you got to try a lot of different things and get positives from a lot of different models and a lot of different tissues before I'm really convinced that it might be real. So
In one of the papers that one of the many papers that you've published, I was looking at the identification of novel mechanisms involved in generating localized vulvodynia pain. And you talked about your tissue specimens in that paper, and I thought it was so lovely that you have always the internal control against the patient's tissue that doesn't have pain, and then also against controls that don't have the painful condition, which seems like you get double the information in terms of comparison and genetic information. So
yes, double the work too, but
yeah,
double the
yes,
information.
yes, yeah. So how many students do you have, or how many
But
graduate students do you have working with you now?
I have 2 graduate students who are part of my lab,
Yep.
one MD PhD student that's been rotating this summer, and I've lost track of the number of undergrads at this point. I think there's
Yeah,
about a dozen of
that's amazing.
that are working at various projects. So we've had a couple of summer students working with us on our Vulvodynia project, which has really been rewarding. One of them's been working with one of my graduate students, the other
Yeah.
one more directly with me. So yeah, we've had quite a few, and we have a couple of technicians in the lab as well.
Okay.
So we've become a very big group. And the thing that always kind of excites me and warms my heart is that a lot of these students are doing this maybe for course credit, but a lot of them are working even over summers for free. And I'm like, I don't know how you're paying rent, but they're just really excited to be doing this work. There's no one doing work quite like this. We love working with you. We love the environment. And we all try to help each other out. So, I believe in the mentality of, I like the little cartoon where there's balloons in the air and everyone's trying to jump to grab them individually. And then you see people kind of getting on each other's shoulders and they're able to reach that goal much easier. So we kind of all help each other out. And since we're in a clinical department, we also do the see one, do one, teach one model.
Yes.
So nobody is, above learning from somebody else or above teaching somebody else. We all help. So if, undergrad that's been in the lab for a month knows how to do something and they're teaching one of the graduate students who's, a year 3 graduate student, nobody gets worried or upset about that. We all just, we know that there's enough external forces that don't support, women's health research and gynecologic basic mechanisms research, that we have to work together if we have any hope of fighting all those forces and actually understanding what's going on. So I'm very blessed. And as my neighbor commented recently, said, oh man, I remember when you first started, you had that one technician in the lab and, it wasn't the best fit. And now he says, you have like a whole army of students. He said, and they're not in there joking around. Like they're in there and there's 3 at the bench over here and 2 over here. And they're just working, very studiously on what they're doing. And I said, yeah, I don't know. Like, I don't even threaten them. I just, they're just.
Well, there's a lot to do.
There is so much to do. So I could keep an army busy for the next, however long I have on this planet.
Yeah. Yeah.
So,
Well, I hope it's a long, long
but
time
it's
because we need
fun.
you.
It's really rewarding.
Yeah. Yeah. Well, I really appreciate you talking with me. I'm going to let you go because I know I want you to get back to all of these really important projects. But I so appreciate it. And thank you for your patience with the technical difficulties.
Yes, well, I'm sorry I'm not a whiz yet, but
No.
It's practice makes progress.
Same. Are you going to be at ISSVD
Yes.
in Slovenia?
Yes.
Okay,
yes.
well, I'll see you there.
Actually, it's going to be a lot of fun because my husband and son are coming too. We're taking a week after to go see Italy.
Oh my gosh, great. Well, yeah, we'll be presenting a couple projects there, one on Ehlers-Danlos and one on medical gaslighting. So I'll be there. Yeah.
Oh, that's going to be fantastic. Yes, I look forward to that. Yeah, I am presenting as well on what's new in Vulvodynia pathogenesis. So, I'll be another summary of some of the things we talked about today. But yeah, I'm looking forward to that trip. So.
All right. Well, very much looking forward to it. Thank you so much again for talking with me. And I'll look forward to seeing you in September.
Okay, take care.
All right. Have a good one.